All right, thank you all for coming. I'm just gonna get cracking here since that seems to be the way things go. So I'm gonna be talking to you today about when animals should innovate and what factors should determine who is an innovator and who is not. So I am working with a definition of innovation that is widely used in the literature. And that's when an animal learns to use a new resource or exploit a current resource in a new way. And I'm emphasizing that first part of the definition because that's what I'm focused on here. So some of the most comprehensive work on adoption of novel prey items has been done in birds. So Lefebvre and colleagues in 1995 did a massive analysis of the literature where they looked for records where birds were recorded preying on things that they had not been previously recorded to prey upon. And this is one of the articles that they cited, an American kestrel preys on least weasel. So this is a dramatization of that very first predation event on a least weasel by a kestrel, courtesy of Dolly 3. But this is something that can have really interesting and compelling importance for issues of conservation. So this is a cane toad being depredated by an Australian swamp hen. So cane toads are these nasty, toxic amphibians that have been invading Australia. And some of the native fauna have learned how to exploit them as a resource. So this is a great story about an invader being adopted into the diet of a native species. But sometimes innovation is just what happens in the day-to-day course of an animal making its way in the world. So octopi are often quite solitary, and so they don't have a lot of opportunities for social learning. So everything that an individual octopus is doing can be thought of as an act of exploring its environment and participating in innovation. So octopi have, at least certain species of octopi, have a hardwired instinct to prey upon crabs. But as some of us learned by watching octopus documentaries during the pandemic, they will eat fish but have to exert a lot more effort to acquire the ability to handle them. So this is sort of a novel prey for them that they, on an individual basis, have to learn to exploit. And innovation, as I've alluded to, is something that can be transmitted socially once one individual has discovered it. So here is some iconic work from Lucy Applin, where she worked with great tits to teach them to use either the blue side or the red side of this puzzle box and just bop the little thingy on one side or the other to get a food reward. So she trained birds to do this in the lab using either blue or red, and then she released them into populations in the wild. And you can see here that populations that were seeded either red or blue saw a really rapid rise in adoption of these novel foraging techniques, but control populations were exhibiting much spottier behavior. And here's that data from a little different perspective. You can see that the number of successful box openings and the preponderance of either blue or red were far lower in those control populations than in these blue-seeded and red-seeded populations. So innovation is, of course, the sort of like the genetic variation, source of genetic variation on which cultural evolution might act. But I'm focusing more on that act of discovery or invention in this talk. So why an individual animal in the absence of social information should attempt to innovate rather than pursuing what it is already familiar with or knows how to eat by one way or another. So there have been different hypotheses put forward in the literature by authors throughout the years, and some of them produce dramatically contrasting predictions. So the excess energy hypothesis is one that puts forth that the more energy you have available to you at the time, the more willing you should be to innovate, because you are risking a negative outcome that is less hazardous for you in terms of fitness loss than it would be for an individual that has less resources to start with. So this might be familiar to people who think about risks that are taken in say, the context of investment. But equally familiar might be the logic behind the bad competitor hypothesis. And this predicts the opposite pattern, which that as you have more and more energy available to you, you should take fewer and fewer risks because you are already able to meet your baseline needs. So this would predict that individuals with the poorest energy reserves are going out there and doing crazy things to obtain them. And there are also some more ad hoc hypotheses, like older individuals might be more innovative, or younger individuals might be more innovative, perhaps due to their correlation with dominant status or availability of energy reserves. And people have predicted correlations with personality as well. So neophilic personality types might be more innovative because they tend to explore new things more and just have more opportunities. And social network position might either provide some social cachet to innovators if they're really well connected, or provide innovators with some buffer against risks of innovation. So many other hypotheses are possible. And Amici et al. Already thought of all this and were like, hey, yeah, let's do a meta-analysis of all the papers that have looked at innovation and see which factors and which hypotheses are well supported. And they did find support for a number of hypotheses and interactions in some cases. Oh, well, maybe not interactions, but the significant support that they found is modified to a large degree by interactions with other things. So they were looking at sexual dimorphism, age, body condition, dominance rank, whether populations were captive or in the wild. And so they found this positive correlation with age, but they also found that that depended on whether an animal was in a captive population or in a wild population. And this inconsistency in a lot of the trends that they found and contingency on interactions really makes it difficult to infer what's actually causing an animal to be more innovative. So this is my entry point where I'm like, we need an ultimate model that could explain why an animal might innovate in a particular context. So I envisioned a really simple system with a single predator individual that knows how to feed on one kind of prey and doesn't know how to feed on another kind of prey. And I included factors of individual state, so how much energy is available to an individual and the individual's age. And then I also incorporated ecological variables, so things that are exterior to the animal but that it could infer from its environment, like the abundance of known prey and the abundance of novel prey. So I used a method called stochastic dynamic programming to arrive at an optimal policy for what an animal should do when it's given the opportunity to innovate on a prey that it doesn't yet know is worth exploiting. So the stochastic dynamic programming is a really great method to use when you have decision-making under risk or uncertainty. So I've drawn up some really simple things here where there's a highly variable outcome for choice A with certain probabilities that differ from a less variable outcome for choice B. And if you do the math, you can see that on average, B has a higher average outcome than A, which is great, so you should choose B unless you have to get 4 resources to survive, in which case you must choose A, because that's the only chain along which you could possibly arrive at an outcome that will keep you going. So dynamic programming lets you do some really fun things that are important for thinking about this kind of decision making. So you can string together a sequence of decisions into a huge chain of events and find optimal policies along that chain of events. You can also apply fitness functions. So for example, in this one, the individual has to have a certain level of resources in order to survive at all, and it might also have some maximum level of resources, at which point it's no longer beneficial to acquire more. And individuals can learn. So as they go along sampling their environment, they can update their estimates about the environment based on whether or not they've succeeded or failed using a particular option. So here is a diagram of the dynamic programming equation that I wrote for an animal that is choosing whether or not to innovate on novel prey. So in a given time step, it may, through a random process, encounter or not encounter a novel prey. So it's not actively seeking it because it doesn't yet know that it's something worth hunting. And if it does encounter this novel prey, which is this toad here, it can either choose to ignore it and go hunt for familiar prey, or it can pursue it. If it pursues it, it can fail or succeed, and on the basis of that outcome, it'll update its information about the probability of this prey being good to eat and the impact on its energy reserves. And there's a corresponding chain for choosing to pursue familiar prey or known prey. So here are the results when I take this predator's algorithm and put it in a situation where it is always encountering novel prey and it is always failing to hunt them. So I'm measuring how many times the predator is willing to keep on trying to succeed with this novel prey, and I call this persistence. And I assume that persistence is directly proportional to the probability of successfully innovating eventually. So what I find is that age and energy reserves have strong impacts on whether or not an animal persists. And in this light shaded region, the animal is attempting to innovate once, and in this dark shaded region, this animal is attempting to innovate twice. So here you might say that young individuals with a lot of energy reserves are going to be the most innovative. But there's also some really weird wrinkles to this output. You'll notice here that there's a nonlinear pattern at energy reserves of 10, where you innovate for part of the time, and then as you get older, you become less innovative, then you go back to innovating, and then you drop out of innovating. And you do something similar when you have energy reserves around 3. So there's this middle-aged tendency to innovate. And there's also what I am calling a middle-class tendency to innovate. Whether or not that terminology is useful, I leave up to you. But starving individuals can't afford to innovate because they risk death, so they're going for the known thing. As soon as they accrue a little bit of energy reserves, they start taking more risks because this is the fastest way to accelerate themselves into an area where they would have relatively high fitness. And then here, your upper middle class individuals are not innovating at all, and it's only these really maxed out individuals that are innovating. So all of these results are further modified by the ecological abundance of known prey and novel prey in the environment. So I just showed you results from common known and common novel prey. But you can see a lot of these nonlinearities persist when rare prey or novel prey are rare. And there's a huge effect of the abundance of known prey. When predators are really at risk of starvation, they become really innovative. So that kind of swamps the effect of the abundance of novel prey. And the big take-home message from this really complicated figure is just all the nonlinearities that are happening. So it means that simple hypotheses like a linear relationship between state and innovativeness can be true in different directions in the same system, depending on what part of parameter space you're looking at. So that could explain why we get these weird results when we look across all published studies. So ideally, in innovation experiments, we would try to control an animal's perception of known and novel prey availability, and at least report individual state relative to the rest of populations. And then we could also experiment in some limited circumstances with some nonlinear models. These results are also strongly parallel with a model by Tom Sherritt and Julie Morand-Ferron on neophobia. So those behaviors seem to have a strong underlying conceptual basis, at least on the ultimate level. This doesn't take into account any social learning or social networks, and it also assumes that individuals have the plasticity to respond. So if a species commonly experiences a certain level of energy reserves at a certain time of life, it might be quite canalized. Okay, I am out of time, so thank you very much, and welcome our next speaker.
So welcome to this presentation on chemical mate choice copying in Drosophila melanogaster. My name is Blue, and I'm a first-year graduate student at the University of Memphis in Memphis, Tennessee, USA. Today, we will explore the fascinating world of how these tiny flies make critical decisions about their mating partners using chemical cues. The study sheds light on a novel behavior in mate choice that has significant implications for understanding sexual selection and reproductive strategies in invertebrates. Mate choice is a crucial aspect of reproductive success, especially for females who often bear higher reproductive costs, such as gestation, parental care, and nutritional needs. Females assess potential mates based on traits like coloration, courtship songs, and pheromonal profiles. Collecting this information can be time-consuming and energy-intensive, particularly in environments with a high density of potential mates. As a result, females have evolved strategies to utilize social information that can reduce the burden of individual assessment. Using social information, females can observe the mating choices of others and copy them in a behavior known as mate choice copying. This strategy has been documented in various species, including birds, fish, mammals, and invertebrates. By observing successful matings, females can make more informed decisions, potentially increasing their reproductive success. Most studies reporting the existence of mate choice copying have relied on experimental setups testing for mate copying via visual cues, even when the model organism primarily communicates through other sensory modalities, such as chemical cues. Chemical cues do not require direct observation of copulations and offer unique advantages that have not been thoroughly explored. Chemical cues are critical in the communication and mate selection of many species, including Drosophila melanogaster. These cues can convey essential information about a potential mate's age, dominant status, and parasite load. Unlike visual cues, chemical signals can persist in the environment, allowing females to gather information about male mating success without directly witnessing the mating event. This study explores the potential for chemical mate choice copying in Drosophila melanogaster, providing new insights into the role of olfactory signals in social and reproductive behaviors. We investigated whether D. melanogaster uses chemical mate choice copying, or the ability to use chemical cues left behind after copulation to choose a potential mate. These social insects form mixed sex groups, offering females a range of potential mating partners. During mate choice, female flies consider a variety of male traits, including body size, age, pheromonal profiles, courtship behavior, nutritional status, and symmetry. D. melanogaster primarily communicates via chemical cues with cuticular hydrocarbon profiles, or CHC, playing a crucial role in attractiveness and mate choice. These profiles are genetically encoded and vary between strains. Several male pheromones, such as cis-vicinal acetate, palmitoleic acid, and C-trichosine, have been identified as key factors in female mate choice. Our research aimed to investigate whether D. melanogaster use chemical cues to copy the mate choices of others. We hypothesized that student females exposed to chemical cues from recently mated teacher females would preferentially choose the same male genotype as the teacher. The study represents the first empirical demonstration of chemical mate choice copying, potentially revealing a novel pathway through which social information influences mate selection in invertebrates. The experiment went something like this. Virgin females are aged 3 to 7 days. Students are collected without CO2 to prevent memory-related issues. Teacher virgins are introduced to a male, denoted as M4 or M7, for a teacher mating phase. Teachers who mate are transferred to an arena with a student virgin, where the student will be exposed to a pheromone profile of the mated teacher. This teacher will have male pheromones from the mating process and the mating plug that is left behind. The student is then transferred to a new arena with our 2 male genotypes, both dyed at random, one red, one green, using fluorescent powders. Ultimately, we divided the females into 4 treatment groups: no teacher, virgin teacher, M4 mated teachers, and M7 mated teachers. This design allowed us to isolate the influence of chemical cues on mate choice copying behavior. Our results indicated that student females exposed to M4 mated teachers showed a significant preference for M4 males, more frequently than those exposed to no teacher, virgin teachers, or M7 mated teachers. This preference was statistically significant and demonstrated that chemical cues from mated females influence mate choices in Drosophila melanogaster. Additionally, exposure to chemical extracts from mated females, but not from unmated males, reinforced this preference, underscoring the importance of female-derived cues in this behavior. Our findings provide evidence of chemical mate choice copying in D. melanogaster. This behavior allows females to utilize residual cues left on mated females to make more informed mating decisions, enhancing the reproductive success. The study highlights a mechanism through which social information can be collected and exploited, broadening our understanding of mate choice dynamics in invertebrates. These insights have implications for understanding the evolution of mating behaviors and the role of chemical communication in sexual selection. Chemical mate choice copying can intensify sexual selection pressures on males, particularly in high-density populations. By increasing the choosiness of virgin females based on social information, this behavior can lead to stronger selection for male traits that are preferred by the majority of females. Understanding these dynamics can help us appreciate the complexities of sexual selection and the evolutionary pressures shaping mating behaviors in natural populations. So in conclusion, our study reveals a novel behavior in Drosophila melanogaster, chemical mate choice copying. This behavior underscores the importance of chemical communication in social and reproductive contexts, offering a new perspective on how females make mating decisions. By understanding these mechanisms, we can gain valuable insights into the evolutionary strategies that enhance the reproductive success and shape the diversity of mating behaviors in nature. I would like to thank you all for your attention and my wonderful research team for all of their help in this work. And if you have any questions, I'd be happy to answer them. Okay, start with you.
So I'm curious, in your experiment, have you tried to see if they can change their mating choices afterwards? Like if you teach them again with a different choice?
We have plans to do that in the future, yes. Okay. So currently, I have no results to give on that, but we are planning on that. Yes?
So you call it the M4 and M7 males, is that right?
That's correct.
Are there particular lines that have a particular chemical profile? Or?
So these are actually lines that we collected Drosophila melanogaster from local Memphis, and then we created 9 isofemale lines. These are the 2 remaining that survived after 20 generations.
Okay. Do you know, have you done the chemistry to see whether the males within the lines have got
Not yet. Hoping that a nice grant will allow me to do that. Any more questions? Yes?
Really interesting talk. Did you notice any differences in terms of copulation vacancies or mating durations in your assays?
What was the first one? Not mating?
Mating vacancies or mating durations?
So I did not notice any. This experiment took some troubleshooting. So in the beginning, outside of our behavioral room, it did take much longer, but once we put them in the behavior room, without any distractions or vibrations or excessive noise, things were pretty normal. Any more? Fantastic. Thank you so much.
Okay, it's just about 3, so I think I'll get started. I'm excited to follow up with another Drosophila courtship talk. My name is Justin Walsh. I'm a postdoc at the University of Pennsylvania in Yoon Ding's lab. And in our lab, we study the evolution of behavior. So when we look at the animal kingdom, we see this remarkable diversity of behaviors. And we know that this behavioral variation is often adaptive. It's often important for species-specific survival and reproduction. But what is less well understood is how genetic changes and changes to the nervous system interact to give rise to the evolution of novel behaviors. And that's really the main focus of our lab. In our lab, we study Drosophila courtship behavior. Courtship in Drosophila looks something like this, where we have this series of discrete, very stereotyped behaviors that include things like males orienting towards the female chasing the female, tapping her with his legs, during which he's tasting her sex pheromones. Males also sing to females, and they produce this song by vibrating one or both wings. And the point that I want to make here is that for all of these behaviors, and really other aspects of courtship that are not depicted here, there's an incredible amount of behavioral variation across these relatively closely related Drosophila species. Just to give a couple examples, there's a remarkable amounts of variation in the sex pheromones they produce, and also in that response to these sex pheromones across species. There's also variation in how important visual cues are during the courting process. So males of Drosophila suzukii, they have these black spots on the tips of their wings, and during their courtship ritual, they spend a lot of time in front of the female doing these wing opening behaviors. And really visual displays like this are much less common throughout most of the rest of Drosophila species. And I mentioned these courtship songs. There's really, again, an incredible amount of variation just across very closely related species. So we think of there as being these two types of song, pulse and sign. And within each of these song types, there's variation in things like frequencies and amplitudes. But then even more dramatically, there's been gains and losses of entire song types. Okay, so given this remarkable amounts of behavioral variation in courtship, what I've been thinking about the last few years is what is going on at the neuronal level to give rise to this behavioral variation across species. So I put together this very simplistic diagram to orient you towards the types of questions that I've been thinking about. So let's imagine we have species A that performs behavior A, and there's a neural circuit underlying this behavior. And let's imagine in this neural circuit there are these three different cell types designated by these different colors. And you can think of these cell types as performing different functions within the circuit. You can think of excitatory versus inhibitory neurons, sensory versus motor neurons, for example. And importantly, it's going to be important later on, these cell types, if we looked at their transcriptome, they're going to be very distinct from one another. Now let's imagine a second closely related Drosophila species performing a similar behavior. We can think of these two different behaviors as maybe two types of courtship song, for example. And in the homologous neural circuitry underlying this behavior, what I've been thinking a lot about is what could possibly be different that produces a divergent behavior in this species. So just what are some of the possibilities? So for example, we could have the gain or a loss of entire cell types, shown here by these blue circles, right? And something about this gain or loss introduces some new functionality to the circuit and drives a slightly different behavior. Alternatively, instead of having a gain or loss of a cell type, we could just have gains or losses in the number of neurons of the types already there, shown by this gain of yellow neurons. So you can think of this as like gaining more excitatory neurons so that the fly may respond more readily to stimulus or something. And then finally, this is meant to be vague, really just meant to encompass anything that is not a change in neuronal number, but we can imagine something like gene expression variation that is functionally changing something about the circuit, leading to divergent behavior down the line. So this is not meant to be an exhaustive list. I recognize that there are other possibilities, but I just want you to consider these three things as we move throughout the talk. So fortunately, we know a lot about the neurons underlying courtship in Drosophila. The majority of these neurons express the sex determination gene double sex. And in the adult male fly brain and ventral nerve cord, there are about 900 double sex expressing cells. And I just want to point out here that we know a lot about what these different neurons are doing. I don't have time to go over these different names and different functions, but I just wanted to point out that we do have an idea of what a lot of these neurons are doing, allowing us to make testable hypotheses if we do see differences across species down the line. Okay, so this question really kind of sums up what I've been working on. How have these neurons, which we know play a role in courtship, how have they evolved across species to maybe give rise to this variation? And to answer this question, we looked at these four species, Drosophila melanogaster, the traditional model system, and then these three sister species here, Drosophila jacoba, Santomea, and Tessieri. And we chose these three species for a number of reasons, one of which is that our lab has had a lot of success transferring genetic reagents initially made for melanogaster into these other three species. That really allows us to do things like label specific neurons and potentially activate or inactivate them, for example. Another reason is that, again, there's just an incredible amount of behavioral diversity even within these three closely related species. There's a lot of courtship differences among them, but probably most strikingly is that the common ancestor of Jacoba and Santomea lost the ability to sing what we call sign song, and instead has evolved a second type of pulse song. So really you have this really striking and interesting behavioral difference. Okay, so to get at this, we took a single-cell RNA-seq approach in which we labeled double-sex neurons in all 4 of these species. We're only talking about males. I'm only talking about males. So then for roughly 100 males per species, per replicate, we dissected out the brain and ventral nerve cord. We sorted out just those double-sex expressing neurons and then followed a traditional single-cell pipeline. Okay, so what did we find? I'm going to show you a number of plots like this, so I just wanted to take a second to explain what they are in case you're not familiar. These are UMAP projections in which each dot in the plot represents a single cell. They're clustered in the two-dimensional space by transcriptomic similarity. And in a second, I'm going to show you a colorized version of this plot. The different colors are meant to represent different cell types. Right, so I already showed you this, all these kind of different spatial clusters, and mentioned that we might know something about their function. So the first thing we really had to do was match the cells in our data sets to the identity of these neurons here. And we did this through IHC staining using some known markers, some Hox genes, and some marker genes that we identified ourselves. And for all of these larger groups, we were able to find them in our data set, which was great. And then from there, we extracted each of these larger groups for further clustering analysis to really get at the heterogeneity within each of these groups. So for example, in this blue group, which is PC1 in the brain, we resolved this out to 6 distinct cell types. So we did that for each of these larger groups. So showing you that for all of these, we found 84 distinct cell types across these 900 double-sex expressing neurons. And here I just want to take a second to really point out how powerful we think our data set is. For example, in the upper left, that square is meant to represent ADN. ADN is a cluster of just 2 neurons on either side of the male brain. And in our data set, we're able to distinguish between those 2 neurons. So we're really excited. We really think that we're going to have a lot of power here to really disentangle what's going on with these neurons across species. Okay, so we looked at kind of what was going on with all these cell types, just really kind of at a more of a broad view across species. And what did we find? So we found of those 84 cell types, 83 of them were conserved across all 4 species. There's only this one species-specific cell type shown here in dark blue. It's a little hard to see. This was a melanogaster-specific cell type. I'm going to talk a little bit about this towards the very end of the talk. But overall, we can really conclude that situations like this, where we see the gain or loss of entire cell types, seem to be very rare, at least in what we're looking at. Alternatively, we also looked to see if there was a species bias in cell number within these cell types. So here I'm only showing you data from 2 of those 4 species, melanogaster and Yakuba, but it's similar no matter what kind of comparison you take. And we found that 11%, 9 of the 84 cell types do show this species bias in cell number. And we're really excited about these cell types. We think that they're really interesting candidates for potentially playing a role in this courtship variation. And then finally, we wanted to get a measure of the transcriptomic similarity across subclusters, across species. So what we did was we performed Spearman rank correlations on the gene expression of all expressed genes in each subcluster across species. And again, this is potentially a little hand-wavy and kind of early in our thinking, but we really see this wide range of transcriptomic similarity across different subclusters. And again, a little hand-wavy, but we do think that these ones that are less similar to each other across species are really kind of merit further investigation and potentially are playing a role. So just to kind of sum up those last few slides, we think that the gain or loss of entire cell types is rare, but we do think we have evidence that some of our cell types are evolving more quickly than others, given that we see these species biases in cell number, and then also given that wide range in transcriptomic similarity. Okay, and then in just the last few minutes, I wanted to return. I mentioned that we had this one subcluster, this one melanogaster-specific subcluster, and we did explore what this was doing in a lot more detail. And I think that this really kind of highlights the power of our single-cell data set as well. So this cluster, this is again just showing it again, just in a different space with fewer neurons. This cluster was the subject of our paper published in Current Biology earlier this year. in which we utilized our single cell data sets to identify genetic markers. This one here is the gene 17D3. Genetic markers that uniquely label this melanogaster specific cell type. And then using that marker identification, we can make genetic reagents to label just those neurons. So that's what you're seeing on the right. And I don't have a ton of time to go over the results of that paper, but ultimately in that paper we show that that melanogaster specific cell type is actually a neuronal cell type called TN1A. And TN1A is essential for the production of sine song. So melanogaster has these neurons, they can sing sine song. But if you'll remember, Yakuba has lost the ability to sing sine song, and in the paper we show that they're also missing these sine song essential neurons. And then we get into a little bit about how that happens developmentally. So if you're interested, I suggest you check out the paper. And then again, so that was really just kind of a deep dive into one of these cell types, right? So there's 83 more. So I just wanted to point out here at the end that we're collaborating with David Chen at NYU to identify just 2 or 3 genes that uniquely, in a combination of 2 or 3 genes, uniquely label each of these cell types. And we're working on building a database so that our lab, or really anyone who is interested in any of these cell types, can better explore their functionality. And I think I have a minute or 2 for questions. So I'll thank my lab, our collaborators, our funding sources, and thank you for coming. Yeah.
Very nice talk.
Thank you.
Do you have a sense of whether or not this melanogaster-specific cell type is ancestral or derived from the melanogaster family?
Yes, great question. So there's the way I presented this is like we definitely have these 84 distinct cell types, right? But if you've done single cell work at all, you know that clustering is a little subjective. I do feel pretty confident about a lot of our aspects of our clustering. What am I trying to show here? But one thing I wanted to show is that if you just cluster melanogaster and tessieri can sing sine song. So if you just cluster those two, you see that tessieri, it seems like, does have these cells. And if we stain for them, it seems like they do, but there's just fewer of them. So I think it's likely ancestral. Yeah. Yeah.
Sort of along the lines of what you're saying with difficulty in clustering and categorizing cell types. Like, do you have so you're using purely differential gene expression to distinguish cell types?
Yeah, we've done a bunch of different things. For a number of these 84, we've actually gone in and stained for some of the markers to make sure that it seems like our clustering is real. So we've done that, and we've done another number of other statistical approaches, which are supposed to give you an estimate of kind of how good your clustering is. We've also played around a lot with just messing around with the parameters to adjust the clustering and then just going in and trying to see if there's good genetic markers. And really this is actually, I think, more on the conservative side. I think there are instances where we could cluster a little bit further and still kind of get, but yeah, those are some of the things we tried. Yeah, of course. Any last questions? Yeah.
Beautiful talk. Is there, do you have any idea, sort of from an evolutionary perspective, of why that subgroup might have lost that extra? It's not like there's certain predation pressures or something that would have led to that?
Yes, that's a great question. So there is a lot of pressure, especially in zones where these species overlap, to avoid hybridization. So we see strong selection pressure to produce species-specific songs. So perhaps it's the result of that. I'm trying to differentiate between tessieri. That's my best guess. Yeah?
Do they gain something to compensate? Like, is there a transformation of lineages?
The lineages that lost?
Yeah.
Yeah, so they actually, they sing a second type of pulse song.
Oh, no, but I mean in terms of cell types.
Oh, in terms of cell types, no, they don't. They just have few, they just are kind of missing this one cell type. They just have fewer of them in that group.
Okay, great. I'll get going. So I'm going to talk today about information, spatial structure, and the coevolution of cooperation and social networks. And this is work done with a former high school student who was working in the group, and he's now moved on to Purdue. So I'm excited for him, but also excited for all the work that we get to share today. So as many of you know, cooperation and other kinds of social behavior are interesting, and in particular allow animals to be able to accomplish tasks and do activities that they otherwise would be unable to do. So we've had a huge amount of empirical and mathematical work that's addressed the question of what are sort of the evolutionary roots and causes of this. And you could sort of, there's many ways to classify them. This is potentially one. Some of these behaviors allow individuals to coordinate with one another to accomplish fitness-relevant tasks. In other cases, they allows individuals to specialize on tasks, allowing a group to perform those tasks more efficiently or to perform more tasks than they normally otherwise would be able to do. Mutual aid is often an important one to solve social dilemmas and other kinds of public goods problems. And then often combinations of these. So this is a great example of these castels in Catalonia, where individuals sort of form these big human towers, much bigger people down at the bottom, much smaller people and children up at the top. So things that sort of involve both of those kinds of activities. And in general, the conditions for this are, benefits of those behaviors from the perspective of fitness have to outweigh the costs, and there usually has to be the right kind of social structure or mechanism in place for these kinds of behaviors to evolve. And those social structures and mechanisms are really sort of like the bread and butter of a lot of the research in social evolution, evolutionary theory and empirical work today. So these can include, of course, things like how do they form affiliative or friendly-like relationships? How are they living in structures that may allow them to interact with kin? How are they keeping track of the behavior of other individuals in the population through reputation or other mechanisms? What I'll focus on today is a specific, kind of very appealing for all kinds of reasons, metaphor for how to track how individuals affiliate with one another are these social networks. These are, of course, these well-known Japanese macaques here, sort of hanging out and grooming each other. Lots of great work now, even using machine learning to be able to automatically capture and quantify these individual interactions and be able to form then social networks from those data. So we're actually getting more and more data for that, not just, of course, for these macaques, but for many different kinds of species. So it's really a rich field, I think, that's been growing at a really rapid and exciting rate. Of course, it derives a lot of what it sort of does in terms of the mechanics and the numerical methods from the previous social network research that's in sociology and political science, studying how humans, another animal, of course, interact socially. So much of the evolutionary theory on this kind of question, particularly in relationship to how behaviors like cooperation evolve on these social networks, there's been a ton of that work, a lot of really interesting work that showed how the structure of that network is really critically important to not just sort of whether cooperation can evolve or not on these networks, but how fast it evolves and other kinds of important issues. But one of the things that's maybe, that connects a lot of that work that we can sort of start to extend from is many of those models assume that the structure of the network is fixed, so it doesn't change over time. And what that means, of course, is that things like the affiliative relationships that individuals have with one another or how related they are genetically to one another, those things are also fixed over time. So we'd like to maybe sort of take a step forward and think more about networks that might change dynamically, or in some sense, maybe even better, emerge from some kind of more organic or biologically motivated process. So I'll talk about one a model that's sort of based on a kind of a model of that process, and this goes back to some work from Amial Elani and Errol Akshag, who's in the room with us today, on a model that was inspired by animal social networks. And really kind of at the core of that is a simple idea that social networks might actually in part be built by the fact that as individuals are born into the social group, that they acquire at some rate the social connections of their parents. They inherit some of those social connections and then form at some other rate social connections of their own. So we call this, or they label this social inheritance. So the idea is that you have a newborn individual here, and at some rate PR, they form a connection to random individuals in the population, and then at some rate PN, they form connections to their parents, to the friends of their parents, the network connections of their parents, and then usually sort of we assume that it's very close to one or exactly equal to one, that they always connect to their parents in the social network. So that sort of early paper was able to sort of show nicely that there were some, even this model, which has very few parameters, as simple as this one, was able to reproduce some or match fairly well the network statistics that you can sort of gather from real or empirical animal social networks. So things like the degree distribution, the distribution of clustering coefficients, that those fit fairly well. And so then, of course, maybe relevant for the thinking about how cooperation evolves on these networks, we like to actually use this network mechanism, and so in combination allow that network to emerge by using these probabilities of connecting, and then see how cooperation and conflict evolve on those networks. So we make sort of standard kind of assumptions we often make in these evolution of cooperation models. There's some benefit that's shared by a cooperator to whoever they're connected to on the network. That comes at a cost for the payoff of that individual who happens to be the cooperator. And then we assume in this particular case that fitness, this just allows us to sort of scale the effective fitness, so it's 1 plus this parameter delta, which measures the strength of selection, and then we raise that to the payoff, whatever they get from interacting with their neighbors and getting whatever benefit they get and paying whatever cost they pay if they happen to be a cooperator. We assume that sort of every time step is a little bit like a Moran model. One individual dies randomly and then is replaced by an offspring of one of their neighbors, and which parent we select to produce that offspring is, again, determined by their payoffs. If there's a mutation, that's sort of normal. So this is sort of previous results. So Errol then went and did a model and showed sort of if you fixed these probabilities of connecting, that if you've got a lot of cooperation. So basically on the x-axis here, you have this probability of linking to random individuals. On the y-axis, you have this probability of linking to your parents' friends. And actually, cooperation was at its highest whenever you had a very low probability of linking to random individuals. So that's sort of intuitive. If you don't want to link to these random individuals, they might be not cooperators. If you happen to be a cooperator, they're going to sort of cheat off of your goodness, and then that's going to sort of defeat cooperation in the population. And here we're just sort of scale that across. It's a consistent pattern across multiple different benefit values. And so what this sort of leads to then is, in effect, once you allow these parameters to evolve, you allow both this probability of linking to your parents' connections and linking to random connections, let those evolve, that you'll end up with a collapse of cooperation, because even if you start with this probability of random linking at a low value, it evolves to higher values, leading to sort of this range where cooperation is at relatively a low frequency. So it collapses, and then one thing that was shown in that paper is that you can rescue it by making linking costless. So initially in the model, there's no cost to being able to form a new connection, a new social link. So if you add some cost to linking, then once linking becomes costly enough, and as long as sort of the benefit, the opportunity of not cooperating is not too high, then you can get pretty strong cooperation evolving in that case with strong linking costs. And what happens in that region is that you have relatively weak probability of connecting to your parents' connections and relatively weak probability of connecting to anyone at all. So what that leads to, in this case, is the degree, the sort of number of connections each individual has. In that region where there's high cooperation up here, you end up with very few connections. So the networks are essentially there's many disconnected components, or many sort of separate components, and they all have relatively low degrees. So very many little isolated networks. Okay, so the question that we sort of started off with is, could we rescue cooperation? So despite this sort of fact that it could be rescued with high linking costs, was there a way to rescue it with low linking costs? Or is there any sort of other way to make this model a little bit more general that we could see that cooperation could be a little bit more robust than maybe initially thought? So one way was that we could maybe make this linking not just sort of agnostic as to who you're linking to, but maybe there's a little bit of information about, could you maybe discriminate if you want to link to some individuals who's a cooperator maybe, but not to link to individuals who are not cooperators, who are defectors. And then as well, we have sort of a sense of structure here, right, with a social network, but that may not be, or it could be, but it could be the same or not really, or not the same to some kind of spatial structure, maybe where individuals are what's determining who mates with whom. So we'll add sort of some spatial structure next and see how that affects cooperation. So first, adding a little bit of information, we can see that if we can now take that probability of connecting to your parents' network connections to your parents' friends and break into two probabilities, whether you connect to your parents' friends who are cooperators and whether you connect to parents' friends who are not cooperators. And maybe that's not a totally unreasonable piece of information to have. You interact a lot with maybe your parents as you're sort of growing up. You're with your parents' friends, and you can sort of get some information about who does what. So if you allow those two things to be separate, then you actually get quite a lot more cooperation over the face space. So even for low, so now this is sort of this old region here for high linking costs. That was sort of what was found before. And then for low linking costs, as long as the benefit is high enough, you end up with a very strong amount of cooperation. And in that region, you end up with very high probability of linking to those individuals who are your parents' friends, who are cooperators, and relatively low probability of linking to your parents' friends who aren't cooperators. And what that results then in is, and as well, you even start to, in some of that region, you connect as well to many individuals who are random connections. And whereas here at the high linking cost, you had many small connected little networks in the whole population, you're essentially with everyone is sort of connected to one another in one giant connected component in this network. So it creates sort of two different sort of solutions. Under high linking cost, many disconnected networks that are small. Under low linking cost, one big connected network, everyone's together. Okay, so we did that for the probability of the chance that you connect to your parents' friends. What if you are allowed to discriminate, get some information about connecting to individuals who are random connections, who aren't connected to your parents? If you can get some information about whether they're cooperators or not, and then discriminate and connect to one and not connect to the other. So in that case, it actually causes another collapse of cooperation by having this extra piece of information about whether random individuals are cooperators or not. We're only sort of left with a tiny region of parameter space under sort of very high benefit and very low linking cost. Can we sort of get any cooperation emerging at all? And then to sort of show what happens to those the evolution of those linking parameters in that region of space, this is sort of the same kind of connected component we were seeing from before, but that high cost region where we had cooperation evolving before, that's completely collapsed. So in some sense now, this information has been poisonous to at least to the stability of cooperation in that region of parameter space. Okay, so I think so we do a little bit with sort of spatial structure, and so what's maybe one of the surprising things about that is that spatial structure, this now is a slightly different plot to show the difference in the frequency of cooperation, that if we allow individuals to sort of connect only to their nearest neighbors on a lattice, that actually reduces the frequency of cooperation in the population, which may have an effect, may have sort of a source in the fact that there's a little bit less the strength of selection on cooperation is a little weaker because we're only comparing at your handful, your neighbor to your left and your neighbor to your right, those two individuals, their payoffs. But in this case, the spatial structure is actually not connected at all to the social network. So if we actually connect the two of them, so we'll sort of skip past here. And if we connect those two together so that you're making links much more less likely if individuals are far away from you, and then you're also then reproducing with individuals who are next to you, then in that case you can actually recover some of that high linking cost cooperative region. Okay, so I'll just sort of, that's what those things say. Go to our conclusion. So knowing the strategies of your partner can be helpful, but only in some cases, only when it's sort of knowing when those individuals who are going to be very close to you in terms of your strategy, because they're probably, if they're friends of your parents, then probably they might more likely share strategies with you. But if you know the whole information about the population, that ends up disrupting cooperation. It's sort of a similar sense that we might have think that well-mixed populations are not conducive to the evolution of cooperation in general. And then finally, that when social structure and spatial structure are local, and they're connected, then that allows cooperation to be enhanced, but when they're disconnected, sort of a different problem. Cooperation has trouble getting off the ground. All right, and if I have probably maybe not any time for questions, but please find me afterwards if you've got any, and thanks for watching.
Hello, my name is Darra. I'm a fourth-year graduate student in the Crianza Lab at Vanderbilt University, and today we're going to trace the evolutionary symphony of birdsong complexity. So learned vocalization is a behavior that's pretty rare in the animal kingdom. Some of the groups that are vocal learners are elephants, whales, seals, people, and birds. Birds have a specific vocalization called birdsong, and birdsong is pretty widespread among the Aussie and Cleve. So what is birdsong? Birdsong is characterized by bursts of sounds called syllables that are separated by moments of silence. And in the songbird clade, song actually varies widely amongst them. It can go from really, really simple songs like the chipping sparrow to pretty complex songs like the great reed warbler. Birdsong is a sexually selected trait, and it functions in multiple things other than mating, such as territory defense and interspecies recognition. And because it's a sexually selected trait, selection favors more complex and elaborate songs. So song complexity can actually be measured in multiple ways. And because we can measure it multiple ways, I'm going to define song complexity here as how elaborate is a song. And so using these specific metrics, we can actually look at differences in these different songs to categorize them as simple versus complex. So as I said before, there's a lot of things that we can look at complexity, but we can't use them in a vacuum just because you may use complexity in one aspect and then you switch that song over to a different aspect and then it completely changes its complexity. So for an example, the common nightingale here, they have one long syllable, but it's really long, so it's really long, but it's not complex in the sense of its unique syllables. But we have here the house wren, it has multiple unique syllables, but it's in a small span of like 2 to 3 seconds. So my question with this is, if we have all these different features of complexity, how do they evolve in relation to the species how do they evolve in relation to the songbird clade? Do they evolve in tandem or do they evolve inversely? My hypothesis with that is there's a suite of characteristics in song complexity that potentially are evolving together, as opposed to they could be evolving in inversely, whereas some will have a trade-off while others don't. So this project is actually a part of a bigger project that kind of looks at how both song complexity and song rhythmicity both play in affecting song evolution. So this is done in conjunction with Jimena Leon, a fellow graduate student in my lab, and she looks at how rhythmicity changes, and I look at how song complexity changes. So in order for us to do this type of work, we actually had to collect the songs, and there's not a lot of data out on these songbirds in general, so we've had to have a database that has about 120 different species of songbirds with their repertoire sizes. From there, we've been to public sound repositories such as Zeno Canto and Macaulay Library, where we are collecting about 20 songs per species. And the criteria for that is they have to at least be 30 seconds long, and it's adult males during their breeding season. So once we actually collect the songs, we can actually process them we parse them into different bouts per species, per individual per species, and then send them through a semi-automatic syllable segmentation and syllable matching system called Chipper that our lab has developed, and we can actually get specific metrics on specific syllables of each bird. Once the songs are parsed and are segmented, I can actually do analysis on them. So I use 4 different metrics to actually look at them. And I'm going to use these 2 example birds as my example for each metric. So first I look at the number of unique syllables. And so that's just how many unique syllables are in each bout. So we see here the dark-eyed junco has 2 and the common nightingale has 6 here. The bout duration is how long each bout is. The switch rate, or the proportion of switching from one syllable type to the next. And then I also do the syllable path length. And how we're calculating the syllable path length is essentially taking syllable level diversity per syllable and extracting specific metrics such as the frequency modulation, the entropy, the goodness of pitch, and we're putting them into a PC space. And with that PC space, we can actually see them in the 2-dimensional space to see if there's differences in those different species. I have an example of the junco, and we'll see in the next, we can actually calculate the path length based on those syllable diversity metrics. So the black colors were now colored with different colors based on the song file of just juncos. And so as we can see here, you can actually trace the path of each song and actually calculate the path length per individual per species. So in order to validate these metrics against all of our other birds, I've actually picked out 6 specific species, and we kind of categorize them based on their unique syllables. So to avoid saying simple versus complex, it'll be species with lower numbers of unique syllables versus species with higher numbers of unique syllables. And these are the 6 that I've picked out and are going to use to verify these metrics. So preliminarily, we've ran them through there, and this is kind of what we found. Just to validate that the unique syllables are a good metric, we see that species with lower repertoire sizes tend to have less unique syllables. That makes sense because they're the same thing. Species with higher syllable repertoire sizes tend to have more unique syllables. When we look at bout duration, it gets a bit more interesting because just because a species has more syllables does not necessarily mean it has a longer bout. And we've shown that with the grasshopper warbler, they are one syllable, so there is no more complexity in terms of unique syllables, but they are longer. When we're looking at switch rate, we kind of follow the same intuition as our unique syllables. Because there's more unique syllables, we do see that species with higher levels or lower levels of syllable repertoire sizes, they tend to have lower switch rates. Interestingly, also with that is there's a big space where the grasshopper warbler is, and that's because it's that big buzz, and there is no switching there. So we don't get data for that. So with this path length that we are using to look at species, we can get a little bit of the nuances between higher complexities because we're actually looking at individual syllables per species. So as we see here, we can actually compare all 6 of the species in these 2 different graphs here. And you can see that the junco and the grasshopper warbler are both really small unique syllables. So they have smaller path lengths, whereas we have the Stronella and the Lucinia both have really high path lengths, which also makes sense because they have high levels of unique syllables. And that's just the raw data of it. Kind of contributes to what we've been seeing, that overall we can see that lower syllable repertoire sizes tend to have, on average, smaller path lengths as well as mean distance between the syllables in their path length. So overall, I took all these metrics and put them in a correlation metrics just to see if they're correlated at all. And intuitively, as I thought, they were correlated to some degree. And so they gave me the idea that as songs are evolving, all songs are evolving to be more complex. All of these metrics are, for the most part, following that same trend. And so, in conclusion with this, we do have a set of metrics that can actually give us some insight into song complexity. And even though that we're not able to see the nuances of complexity between middle and high complexity levels, we are able to see the comparisons between low and high level songs. And finally, these specific acoustic features are positively correlated and appear to be showing that when we have more complex songs, they all kind of evolve in tandem. So my future plans with this is to actually fine-tune my PCA analysis for the path length by adding frequency modulation and amplitude modulation, taking all of that together and then actually running it on all 120 species that we have remaining, and then combining that data with the rhythmicity data to see if there is any trade-offs between song complexity and song rhythmicity. And with that, I want to say thank you to my lab, and have any questions? Yes.
Yeah, great talk. This was cool. I was wondering, is there any particular acoustic trait that's driving the complexity that you're observing, or have you noticed anything in that complexity?
So that's a complex question for complexity, mainly because complexity is really hard to get at of what complexity is. So in the field, it's normally the number of unique syllables, but that doesn't necessarily mean that that's what's causing it to have the other metrics do it. So it's more of a, we still need to understand what complexity is to see like what's driving it.
Yeah, I was thinking the traits of the syllables itself. But yes.
Oh yeah, no, we don't know that yet.
That's fair. No, that's fair.
Yes.
You might have said this in the beginning and I missed it, but you have, your song is made up of potentially different syllables, but birds often have different songs for different circumstances. How did you handle which song you're looking for? Did you somehow compile the different types of songs?
Yeah, so that was kind of the first thing we had to get over. So how we've narrowed it down to is we just did mating songs during their breeding season. And again, we're relying on community science data, so it's based on their classification of what they have said is that song during that breeding season.
Do you think you would find similar results if you looked at, for example, territoriality songs rather than mating songs?
Yes, I would. I won't definitely. I would. In my analysis of this, we find I also work with zebra finches, and they tend to have a little bit of a different song because it's directed song towards their mate than like when they're practicing or when they're modifying their song. Yes.
Really nice talk. In terms of defining those path lanes from the VC spacing, do you have any kind of notion of how much is going to be affected by building that space from lots of different species with higher ranges of variation versus lower? Because really that's agnostic in terms of the kinds of variation you're capturing and just kind of maximizing and also variance. So do you think that will really change whether that corresponds to your intuitive notion of what complexity means? You're just jumping around that space.
Yeah. So my hope is once I put more species in, I get to see more clusters of, more specifically in terms of higher level complexity. When we picked the higher level unique syllables, we didn't get all the song types, so there's like certain birds that have like 300 different unique syllables. So I would hope to see that once I add more, that I can see clusters based on more species that are like themselves, if that makes sense. Yes?
Have you thought of like including some sort of measure about like entropy songs and harmonic structure?
Yeah, so my PCA, sorry, I kind of went through it. It actually pulls out entropy, frequency modulation, goodness of pitch, and I think I'm missing something. But we do have a script that actually writes out for 3 different types of entropy. And we find that the spectral entropy and Shannon entropy are pretty tightly correlated. And then the odd one out is Wiener entropy. So we have played around with a lot of different entropies of each syllable. Any more questions?